Hello, thank you for joining us for our monthly webinar. This month, we are very pleased to have internationally recognized speaker, Dr. Michael Hyman. Dr. Hyman is a professor in the Department of Plant and Microbial Biology at North Carolina State University, and he has over 30 years of experience in the characterization of microbial degradation processes, looking at environmental pollutants like chlorinated solvents, ethers, and fuel oxygenates. Many of you are familiar with Mike. He's an ardent researcher, a well-known, world-renowned expert in the biodegradation of fuel oxygenates, and I've worked with Mike for more than 15 years, and so he's really the resource of who I go to with questions about these particular topics. I think you're going to find this to be a very useful webinar today. He's going to be talking for us about aerobic propane oxidizing bacteria, kind of explain a little bit more about the processes they're involved in, how they work, and then talk to you a little bit about the molecular tools that we can use for their detection. Now, as many of you know who've attended our webinars before, we are recording this webinar today, and it's going to be available on the resource section of the Microbial Insights website within the next week. No need to contact us. A link will be emailed to all registrants as soon as it's available, so we'll automatically send that to you if you registered for the webinar. And at the end of this webinar today, there's going to be a question and answer period. So if you have questions during Mike's talk, go ahead and go into your GoToWebinar software, click that question section, go ahead and enter that in. And then at the end of the webinar, Mike will go in and will answer as many of those as we have time for. And then if you think of other questions after, you can feel free to email Mike about them, or you can also email us at info@microbe.com, and we'll get those questions answered for you. So without any further delay, I'll go ahead and turn it over to you, Mike.
Great. Thanks, Dora. Thank you all for turning up today, virtually online. I'm doing this from home, and hopefully the dog won't bark during this presentation, but if you hear a woof in the background, please understand what's going on. So as Dora pointed out, I'm going to talk to you about propane oxidizing bacteria and what they do, how they work, and how to detect them. And this is sort of a little bio sketch for myself. I'm a professor of microbiology, as Dora mentioned, at NC State University. I got my bachelor's and PhD in the UK, so you can perhaps tell that I'm originally from the UK from my accent. I did postdocs at UC Riverside and Oregon State. And I've listed here my research interests, and frankly, this is really the research interests that I've listed here, dealing with gas utilizing bacteria, co-metabolism, enzymes and pathways, etc. is kind of going to be reflected in the composition of the talk today. There's a pixelated picture of me from 15 years ago, which I like much better than the one that's available now. And I was sort of noted on, as I was putting this slide together, well, it's actually an old slide, that actually all three of the organisms that I have there on the right-hand side are one way or another propane oxidizing microorganisms. So up here we have Nitrosomonas europaea, which is an ammonia oxidizer. This is a culture of Mycobacterium vaccae JOB5, which is a propanotroph. And then this is a fungus that we also work on. Anyway, after that introduction, I'll just give you a little outline about what we're going to talk about. I want to give you a little bit of a historical context about some of the activities and the uses of aerobic propanotrophs, propane oxidizing bacteria. We're going to sort of segue then into looking at propane oxidation, looking at some of the pathways, and in particular an emphasis on the monooxygenases, because these enzymes are very key to understanding what these microorganisms can do and also underlie a lot of the detection methodologies. One of the things that is going to come up that hopefully will do some reasonable justice to is that it's very clear now that certainly in a lot of propanotrophic bacteria, there are actually multiple monooxygenases, many of which can be concurrently expressed and present. So deconvoluting what those enzymes are doing represents something of a challenge. And then that's sort of going to segue then into the detection methodologies and some of the caveats that you need to think about. Just for the sake of today, I thought I'd give you a couple of definitions. A propanotroph, as far as I'm concerned for today, is an aerobic bacterium. It could also be a fungus, I guess, but we're primarily focusing on bacteria here that can utilize propane as a sole source of carbon and energy for growth. And I put a question mark after carbon, partly because a lot of propane oxidizing bacteria seem to assimilate carbon dioxide. So propane would not necessarily be the sole source of carbon for these organisms. Following on from that, these organisms may or may not utilize other volatile alkanes as growth supporting substrates. So as hopefully you'll see that propane oxidation, there are a spectrum of activities that can potentially account for these organisms and what they do in the environment. And then certainly I think because of the likely audience today, that we've focused on the environmental relevance of these microorganisms, in particular as it deals with the biodegradation of environmental contaminants. I've put a working definition of co-metabolism here, which is something that we're going to talk about. And I'm defining it as the ability of microorganisms to grow on specific primary substrates, and particularly in this case propane, and then concurrently or subsequently biodegrade compounds, otherwise known as co-substrates, that they do not or cannot utilize as sole sources of carbon and energy. So hopefully that will be helpful for guiding you through some of the slides later on. So let's start in very, very general terms, have a look at where we find propane. The atmospheric concentration of propane is roughly, it was in the part per trillion level. Certainly one of the interesting features of the last few years is that the concentration of propane and other gaseous hydrocarbons, volatile hydrocarbons, has increased quite dramatically, as this paper sort of alludes to the title over here. And this is attributed primarily to the production of oil and gas from hydraulic fracturing processes in shale deposits and other deposits. This is sort of a diagram here from the EIA, demonstrating where the major production sites for natural gas and are in the United States, primarily down here in Texas, Louisiana, and up here in the Northeast. This gives you an idea about how dramatically production of gas has increased over the last sort of 20 years. Again, this is data from EIA, and we are currently looking at around something in the region of 50 billion cubic feet per day of shale gas production. And clearly the Marcellus Shale area over here, which is over here, represents a major source of gas. And in fact, this is localized in some of the measurements that were made in this study here. We also find propane a lot at sea, and this is one thing that we're going to find comes up quite frequently in some of the discussions today, is that we're focused a lot on terrestrial propane oxidizing systems, but propane actually has a very, very important role in some of the processes that occur in marine environments. So over here on the left-hand side, we have some of the natural sources of hydrocarbons in the marine environment. We've got things from asphalt volcanoes through to hot gas emitting chimneys. The ones that I think are perhaps most important for propane are the gases that get released from cold hydrocarbon seeps. And this is a very interesting study that came out recently that indicated that organisms, propane oxidizing organisms, or organisms that can grow on short chain alkanes of the genus Cycloclasticus, form symbionts with a lot of deep sea mussels and sponges, etc. And this is a sort of a submersible photograph of some of these organisms around these cold hydrocarbon seeps. And this is a fluorescence in situ hybridization showing the presence of alkane oxidizing bacteria in the gills of these organisms. Propane is also associated with some of the anthropogenic sources of petroleum hydrocarbons in the environment. And one slide that I think is particularly interesting was this one that came from the Deepwater Horizon spill back a few years ago. There's a picture of the burning rig. I'm not sure how many of you have followed that story particularly carefully, but one of the observations that was made, there is a deep sea plume at roughly 1,000 meters below the surface, which contained a lot of the more soluble hydrocarbons that were released from the oil and gas that came from that well. And one of the early studies that came from this very, very intensely studied process was that propane respiration may well have been a very important preliminary process in starting the biodegradation of the hydrocarbons that occurred in that deep plume. And this little dot here gives you an idea of just the concentration of propane within the gas and the oil that was released from that process. When we move on to land, we find propane's kind of important for things like oil and gas prospecting. So hydrocarbon seeps that occur in the marine environment also occur in the terrestrial environment, and they can have impacts on vegetation. As you can see here, we have this sort of area where there is no vegetation, and this is primarily due to anoxia. There is a ring of kind of high productivity from plants, and this may be associated with nitrogen fixation by gaseous hydrocarbon oxidizing bacteria. And then there's this sort of return to background vegetation. One of the approaches that has been used for oil and gas prospecting is to simply plate out soil samples that are obtained from environments impacted like this, and then use plots of the number of colony forming units of propane oxidizing or butane oxidizing bacteria to use this to localize underlying gas deposits. This works primarily on the fact that gases such as ethane, propane, and butane are kind of unique to oil and gas as opposed to biogenic methane, which would obviously confuse this issue. Historically, some of the uses of propanotrophs go back to the use of propane and natural gas for producing things like single-cell protein. Perhaps the most notable one, although not really connected with propane, was the protein process that ICI had in place during the 1980s. This was an enormous process that was used to convert methanol into animal feed protein using Methylophilus methylotrophus. This is the original reactor here. I think it was 1.5 million liters. Clearly, sort of a decade later, ICI didn't see much value in this process and demolished the reactor. But the idea of using hydrocarbon oxidizing bacteria for the production of protein certainly has led to the isolation of several of the organisms that we'll talk about today. Some of those include butane oxidizing bacteria like Tara butanovorans, which has a butane monooxygenase, but also grows on ethane and propane. And then a fungus that we've worked on, Graphium, which grows on C2 to C5 alkanes present in natural gas. Another major interest of using propane oxidizing bacteria was for the production of bulk chemicals. Exxon in the 1980s had a very great interest in using these organisms for making both epoxides and ketones. And the idea behind this is that if you have organisms that are growing on propane and then you supply them with compounds like propylene, that the enzyme systems that these organisms express to oxidize propane can then convert propylene to propylene oxide, which is a useful starting material. And then also the ability to produce ketones. So in this case, you have a monooxygenase that is taking a propane oxidizing monooxygenase that is taking propane, converting it initially to 2-propanol, and then the secondary alcohol dehydrogenases present in these organisms then convert those alcohols through into ketones, which can be, again, used as chemical bulk products. And also some of the other organisms that were isolated that are now kind of relevant for propane oxidation were organisms that excrete acid. So one of the organisms that we've worked on quite a lot recently is a Rhodococcus species that grows on gaseous alkanes. The whole idea of co-metabolism and the role of propane oxidizing bacteria and the ability to degrade environmental contaminants was kind of cemented, I think, by J.J. Perry, who also used to be at NC State University. This is one of the studies that he conducted. And they're particularly interested in the fate of cyclohexane, which is one of the more slowly degrading components of gasoline. And Mycobacterium vaccae JOB5 was originally isolated on a branched hydrocarbon, isopentane. It grows on an enormous range of alkanes, C2 to C20. And one of the notable features is that once it's grown on propane, it has the ability to degrade cycloalkanes. And if you have a look at this little pathway over here, JOB5, when we grow it on propane, has the ability to take cyclohexane to cyclohexanol and then to cyclohexanone, but then does not further oxidize that compound. And Jerome Perry put together an artificial mixture where he took cells of JOB5 plus cyclohexane plus an organism that was able to metabolize cyclohexanone to CO2, and then demonstrate by adding both organisms together that you would be able to get mineralization of cyclohexane all the way through to C14CO2. So clearly an interaction between these two organisms, one of them catalyzing the propane-dependent degradation of cyclohexane, the other one, strain CY6, utilizing the products of that reaction. So if we look at some of the more conventional propanotrophs that are out in the literature, this is a sort of very, very 100,000, well, a 10,000 foot overview of the type of compounds that these organisms can biodegrade. This sort of summary is certainly not intended to be comprehensive, but it is based on the literature, primary literature, as opposed to perhaps the secondary literature. And these are the organisms listed down here. So there's Graphium, the fungus, and then all of these are well-known propanotrophic bacteria. There's JOB5, there's Rhodococcus jostiar, HA1, etc. If you look at the literature that's, the published literature, this gives you an idea of the type of compounds that these organisms are currently known to biodegrade. Chlorinated solvents clearly known to be biodegraded by JOB5 after growth on propane. The absence of tick marks here is not meant to suggest that these organisms do not biodegrade the TCE, but there isn't any clear evidence in the primary literature to report that. A lot of these organisms have the ability to oxidize MTBE, and an even larger proportion of these organisms have the ability to oxidize another ether, 1,4-dioxane. And particularly this series of organisms here also has the ability to degrade NDMA. A couple of caveats that I would put into this. Again, not all of the co-substrates either tested or listed, so this must be viewed as a sort of 10,000 foot overview. And I would say that the tick marks that you see here really don't pay any attention to the type of concentration that the organisms were exposed to these co-contaminants or the incubation time. And so I think that covers a lot of sins, and this is a theme that I will come back to a couple of times later on in the presentation. The propane utilizing bacteria have been used for the biodegradation of contaminants both ex situ and in situ. I have chosen here to focus primarily on the in situ work. And there are a couple of sets of literature that would be worthwhile looking at. The use of single well push-pull tests for site characterization has made use of propane oxidizing bacteria and stimulating propane oxidizing bacteria. And there are a series of studies that came out of Oregon State and Lou Semprini's group where the idea behind this is that you are introducing into a single well a solution that contains both a reactive and a conservative tracer. Often there is a biostimulation phase where, for example, adding in propane prior to this. Once you've developed a propane oxidizing community, you can interrogate that activity by putting in this reactive and conservative tracer into the well. The substrates move out, you have a resting period, and then withdraw the tracers back and then look at the changes in both the conservative and the reactive tracers and the products that get generated. So this has been done with using ethylene and propylene as surrogates for some of the contaminants that they're interested in, things like cis-DCE and TCE. Propane's also been used in field scale demonstrations. So this gives you some examples here. Back in 2001, there's an ESTCP report describing the co-metabolic degradation of TCE using a combination of propane and air sparging. And more recently, perhaps not more recently, over a 15-year period roughly, Envirogen, which then became Shore, which I think then became CB&I, which is now Aptin. That group was involved in using propane biostimulation and bioaugmentation for both the degradation of MTBE, and that's described in this report here, an EPA report, and then 1,4-D is a paper that was recently published in Groundwater Monitoring and Remediation. In both cases, using propane and biostimulation propane biostimulation and bioaugmentation with this particular strain, Rhodococcus ruber EMV425. I'll put in a plug for a paper that hopefully will be coming out very shortly, which looks at propane stimulated degradation of 1,4-D and chlorinated solvents using a recirculation well. This is Jacob Chu's work from Haley and Aldrich, and hopefully that will be published fairly soon. Again, another example of using propane to biodegrade some of these environmental contaminants. So if you have a look at the type of organisms that are considered as propanotrophs, if you look at the terrestrial genera, most of these are mesophilic. In other words, they're growing at moderate temperatures, and they are all typically facultative hydrocarbon oxidizers. The gram-negative species include strains of Pseudomonas, Variovorax, and as you will see a little bit later on, methane oxidizing organisms of the genus Methylocella. But it's actually dominated by gram-positive strains, strains of Arthrobacter, Mycobacterium, Rhodococcus, etc. If you switch over into the marine genera, these are often psychrophilic organisms. These are organisms that are being pulled out at depth from the ocean, where the temperature is often close to freezing, around 4 degrees centigrade. Some of them, unlike the terrestrial genera, are obligate hydrocarbon oxidizers, and these are some of the gram-negative strains, Cycloclasticus, Colwellia, etc. What is kind of curious is that there seem to be relatively few examples of gram-positive organisms in the marine environment that are recognized as propanotrophs. So if you have a look at the propane oxidation pathways, this I recognize looks kind of complicated, but really the simple way to look at it is that there are two mechanisms or two key reactions that, that influence propane oxidation. The first reaction is the oxidation of propane to 1-propanol by a monooxygenase enzyme. So anything that's listed in red here is a monooxygenase enzyme. And this goes through a kind of conventional hydrocarbon oxidation sequence of going from an alkane to an alcohol to an aldehyde through to an acid. And clearly there's very, very little variation in the subsequent metabolites that get generated from this system. The other mechanism is subterminal oxidation, and this underlies why there's been so much interest in propane oxidation, because this is really the first alkane in the homologous series where subterminal oxidation is possible. And so in a lot of instances, we find that propane gets oxidized to 2-propanol, again by a monooxygenase reaction, and then that alcohol is then oxidized by dehydrogenase to give us acetone, and then there are myriad mechanisms for the degradation of acetone following from that initial monooxygenase and dehydrogenase conversion. Some of them involve the fixation of CO2 and the formation ultimately of acetoacetate and back and forth from storage compounds like polyhydroxybutyrate. There is a Baeyer-Villiger monooxygenase catalyzed oxidation of acetone to methyl acetate, which then spontaneously undergoes hydrolysis to acetate and methanol. And then acetol itself, which is a hydroxylated product of acetone. There are a variety of different mechanisms that can, and some more likely than others, to lead to the further degradation of that compound. So subterminal oxidation and terminal oxidation really is this key issue up here, whether you go through this route or you go through this large expanse of different potential reactions that follow the production of 2-propanol. So by focusing on the monooxygenase enzymes, I have tried to put together a comprehensive list of the enzymes that are known to oxidize propane, the organisms that they're found in, the type of abbreviation, and the type of cofactors that, or type of enzyme that they are, as well as the substrate range, and some indication of whether this is a metabolic or a co-metabolic process. So anything that you see in red down here represents a co-metabolic process and requires the presence of a primary substrate. And there are, to my reckoning, 3 examples of this. One of them is ammonia monooxygenase, which you find in the ammonia oxidizing bacterium. Requires the presence of ammonia to power this, but they can oxidize all of these alkanes. There is a particulate methane monooxygenase, which allows the methane oxidizing bacteria to grow on methane, but also has the ability to degrade some other N-alkanes. And then the last one that we have here is, well, we have 2 others. We have the soluble methane monooxygenase, which does the same thing as particulate methane monooxygenase, oxidizes methane, and then can co-metabolically oxidize these alkanes. And then this odd example of butane monooxygenase, which we find in this organism tara-butanovorans, which will oxidize methane but will not grow on it, but the organism can grow on all of these other alkanes. Everything else that you see in green represents organisms that, or enzymes found in organisms that can otherwise grow on propane. So we're going to walk our way through these bit by bit and give you a little bit more information as we go through. So we're going to do these in order and start up here with graphium and cytochrome P450. So this is an organism, it's a filamentous fungus. It was originally isolated at low pH using natural gas, and the idea was that potentially natural gas and primarily the methane component could represent a source of energy for this organism. It actually turns out that it grows on the trace components of natural gas, so the wet components of natural gas, so ethane, propane, butane, N-pentane, etc. This organism actually grows on a pretty wide variety of interesting compounds, not only alkanes, but also diethyl ether and tetrahydrofuran. It will slowly co-metabolically oxidize other ethers like MTBE and 1,4-D after it's being grown on alkanes. And RNA interference studies with this organism suggest that there's this one particular unique cytochrome P450, which is responsible for initiating the oxidation of all of these compounds. There's the cytochrome P450 right in the middle there. There is the heme group and the iron right in the middle. So this is the kind of structure of these type of enzymes. There's a lot of interest in having organisms or enzymes that can catalyze the oxidation of these smaller alkanes, methane, propane, ethane, etc. And certainly a lot of effort has gone into engineering and evolving P450s that can oxidize things like propane. If we move along, the next enzyme that we're going to look at is alkane hydroxylase. We've done a bit of work with Pseudomonas putida GPO1, which has a very well characterized alkane hydroxylase, which is on a plasmid, the OC plasmid. It is the hydroxylase component of this enzyme, the bit that actually catalyzes alkane oxidation, is a membrane-bound hydroxylase component, and this is kind of its structure here. We have this ring of 6 transmembrane helices. Traditionally, this organism was thought to grow on liquid alkanes, C5 through to about C10, and but it actually will grow somewhat slower, more slowly on both propane and N-butane, but it does not appear to oxidize either ethane or methane. In terms of its relevance, this enzyme we know will oxidize the methyl ethers like MTBE and tertiary amyl ether, but currently there's absolutely no evidence that this will oxidize any of this chlorinated compounds, TCE or 1,4-D. And it should be pointed out that the alkane hydroxylase system in strain GPO1 is while it's been very well researched, it is kind of unusual. We find a lot of other alkane hydroxylases distributed, very, very widely distributed in gram-negative and gram-positive species, but their substrate range tends to be longer chain alkanes in the range of C10 to C18. So somewhat of an unusual example, but clearly a subset of alkane hydroxylase expressing bacteria can grow on propane. Next enzyme we're going to look at is ammonia monooxygenase, otherwise known as AMO. We find this organism, this enzyme in autotrophic ammonia oxidizing bacteria, which are often referred to as AOB, the model organism being Nitrosomonas europaea. But we also find it in other ammonia oxidizing organisms, and these include ammonia oxidizing archaea, AOA, as well as the more recently discovered Comamox strains, which have the ability to oxidize ammonia all the way through to nitrate, rather than the traditional split process of having ammonia oxidizing bacteria or archaea oxidizing ammonia to nitrite, and then a separate group of nitrite oxidizers oxidizing nitrite through to nitrate. So AMO catalyzes the very initial reaction in ammonia catabolism, converts ammonia through to hydroxylamine, and the organism then grows on the electrons and energy released from hydroxylamine through to nitrite. It's a very, very versatile enzyme. We know that it will oxidize alkanes from C1 up to C8, so it will actually oxidize methane. It epoxidizes and hydroxylates and alkenes. It will also oxidize aromatics, ethers, carbon monoxide, and a range of chlorinated solvents. When you look at propane and butane oxidation, one of the nice things about this organism is that it does not further oxidize the alcohols that get produced from these reactions. So you can look at the stoichiometry of terminal to subterminal oxidation, and vast majority of the oxidation of at least propane and butane gives subterminal oxidation products. So isobutanol, isopropanol, 2-propanol dominates over 1-propanol, 2-butanol dominates over 1-butanol. The cofactor in this enzyme is copper. And although the enzyme hasn't been purified, a lot of the evidence supporting this comes from the fact that very potent copper chelators like allylurea will very, very potently inhibit ammonia oxidation and the transformation of all of these aromatics. The organism has no ability to grow on these alternative substrates, so we have a really nice definitive assay there. And the enzyme's also inactivated by light and by alkynes. So alkynes act as mechanism-based inactivators of this enzyme and very potently inhibit the oxidation of both ammonia and all of these substrates. Another enzyme that can oxidize propane is the particulate methane monooxygenase, which you find in methanotrophic bacteria. Again, this is thought to be, and there's very strong evidence, both from crystallography, physiological studies, etc., that this is another copper-containing monooxygenase. And structurally and catalytically, it is quite similar to AMO. It is widely distributed in methanotrophs, although as you'll see in a later couple of slides, in a few minutes, that the traditional view that this was found in all methanotrophs clearly has gone by the wayside. Like AMO, this enzyme catalyzes primarily subterminal propane oxidation. It has a more restricted substrate range than AMO, so it only oxidizes alkanes up to about N-pentane, but it does oxidize some alkenes and chlorinated compounds as well. The structural similarity and the catalytic similarity between these two enzymes, again, is supported by the impacts of copper-chelating compounds like allothiourea and the inactivation of the enzyme activity by alkynes, which act as a mechanism-based inactivator. Certainly one of the more recent discoveries that relates to both AMO and PMMO is this discovery of this enzyme, which is referred to as another CuMMO, a copper-containing monooxygenase, is this particulate hydrocarbon monooxygenase, which I'll talk about in the next slide. This enzyme is designated PHMO, is somewhat related to PMMO and AMO. It's got the same operon structure, so these are the three subunits, the genes encoding the three subunits of all of these enzymes, so this kind of unusual sequence of CAB. It was originally identified in an organism called Microbial CF8, which has the ability to grow on a wide range of alkanes from C2 up to C16. This enzyme was referred to as particulate BMO, PBMO. The activity of that enzyme is inhibited by things like allothiourea, acetylene, and light. And these characteristics were used to implicate the enzyme in the oxidation of a subset of the alkanes here. So in the C2 to C4 range, other enzymes are thought to be responsible for the oxidation of the longer chain alkanes. There is a well-characterized enzyme, Mycobacterium tubuense NBB4, where this enzyme has been cloned, and the same activity that was inferred from the physiological studies with the cardioides CF8 were confirmed with the clone enzyme. It's shown that that enzyme can also oxidize ethene and both cis-12-DCE and 12-DCA. And it's interesting that a very closely related enzyme has recently been demonstrated or can be detected in the genome of a 1,4-D degrading bacterium, Mycobacterium dioxynitrophicus PH06. Certainly the evidence at the moment is that the majority of the detections of this enzyme have been in marine species, and this has come mainly from metatranscript analyses where you're looking at the transcriptional profiles of complex communities. So the next group of enzymes that we're going to look at are the soluble diion monooxygenases, and I'm sure you've heard of this term before. It's been abbreviated to SDIMOs. These are multi-component enzymes with varying subunit compositions, but at heart they all contain a hydroxylase component or components that are responsible for actually catalyzing alkane oxidation. There is a reductase which transfers electrons from NADH from conventional carbon metabolism within the cells. And then frequently there is a regulatory component that can modulate the activity of the hydroxylase. They are a collective group of organisms, of enzymes, in that they all contain this non-heme diiron center in the hydroxylase component. The vast majority of them, and I would say almost now universally, is that they're non-essential enzymes. There's some debate about that. Well, obviously some question about that when we deal with soluble methane monooxygenase. But in all other cases, these are viewed as non-essential enzymes. So the organism makes use of these as a facultative trait. Many of them are plasmid encoded, and that leaves the potential of horizontal gene transfer from one organism to another. So they initiate the catabolism of diverse alkanes, and they get differentiated by the gene operon organization, and then named by their substrate preference. So we're having a look here at tetrahydrofuran monooxygenase. There is the hydroxylase component, there is the reductase component, and there is the regulatory component. So if you look at the propane oxidizing soluble diiron monooxygenases, these are the 6 groups that are currently recognized. Anything in red has the ability to oxidize propane, and this includes methane monooxygenase, butane monooxygenase, and then 2 classes of propane monooxygenase, one of them which is well established, one of them which is much more recently discovered and has been named various different names. Soluble monooxygenase is kind of a holding pattern, and what we're going to call it for today is a short chain alkane monooxygenase, otherwise known as SCAM. The methane monooxygenases are group 3 SDIMOs. The purified enzyme from some of the model organisms can oxidize C1 to C8 alkanes. Propane mainly oxidized to 2-propanol. You can find soluble methane monooxygenase both in obligate methane oxidizing bacteria. Most methanotrophs have PMMO, and may or may not have soluble MMO. There are now examples of obligate methane oxidizing bacteria like Methanoferula, which only contain SMMO. There are some facultative methanotrophs that contain PMMO and may or may not contain SMMO. But the big change in the methane oxidizing world, I guess, is the discovery of organisms like Methylocella sylvestris, BL2, which actually contains both SMMO and a propane monooxygenase, which is a group 5 soluble diiron monooxygenase. So this enables this organism as a facultative methanotroph to grow on methane, propane, or both of those things concurrently. So while these organisms were originally thought to be kind of restricted to acidic environments, it turns out that actually they are really quite widely distributed. Butane monooxygenase, very closely related to SMMO, found in tara-butanovorans. This organism grows on C2 to C9 alkanes. If you look at the purified enzyme, it oxidizes propane primarily to 1-propanol. And there is, well, we don't detect this enzyme frequently in the environment. There is some thought that certainly in marine systems that it might be responsible for propane oxidation and potentially cycloalkane oxidation. We've talked about strain MBB4 briefly. This is an enzyme that has been located in this organism by Nick Coleman's group. They've now expressed this enzyme and shown that it will oxidize ethylene, ethane, and propane, although ethylene is the preferred substrate here. Will also oxidize chlorinated solvents like 1,2-DCA and vinyl chloride. It actually has the same substrate range, or a very similar substrate range, to this particulate hydrocarbon monooxygenase. And there's some thought that this might, this sort of catalytic duplication might be a reflection of the need for copper for PHMMO versus the lack of copper needed for this soluble monooxygenase up here. Propane monooxygenase, as it's most widely known, is a group 5 soluble diion monooxygenase. It was originally identified in this organism, Gordonia ty5, and it has the ability, this organism grows on a wide range of alkanes, but they tend to be longer chain alkanes, and its ability to grow on propane, but not, for example, ethane or butane, really stands out. And this theme gets observed in a lot of instances amongst the propane oxidizing, amongst propane monooxygenase expressing bacteria. And that's what I mean by propane only. It has this ability to sort of focus in simply on propane rather than a broader gaseous alkane substrate range. It catalyzes a subterminal oxidation of propane, so it's otherwise known as propane 2 monooxygenase. The operon, the genes occur in this order, A, B, C, D. There is a similar enzyme that you find in Mycobacterium strains, which was discovered based on its ability to oxidize phenol to hydroquinone. The issue about this enzyme is that it's found in many bacteria, including a lot of nitrogen fixers, which really have no evidence whatsoever. This may be a lack of looking, but no real consistent physiological theme that have the ability to oxidize propane. So there is some question about what the role of this enzyme really is. And certainly when you look at its biodegradation capabilities, the ability of organisms that express this enzyme to biodegrade contaminants, there really is only concrete evidence for the ability to oxidize NDMA. There seems to be little evidence for the degradation of other contaminants. One of the interesting things about it is that when you express this comparable enzyme in E. coli, it has the ability to oxidize acetone to acetol, which you may remember, or you could be forgiven for not remembering, is one of the steps in the secondary propane, the subterminal propane oxidation pathway. And although I don't think Paul Hatzinger would necessarily agree with me that propane monooxygenase oxidizes MDMA by this route, the similarities between MDMA and propane and the reactions that are known to be involved by some monooxygenases in MDMA degradation perhaps bear some looking at. We also find propane monooxygenases, these PRMOs, these group 5s, in a lot of apparently non-propanotrophic bacteria. Example of this would be Methylibium petroliphyllum, which we know biodegrades or grows on MTBE. So you see upregulation of the propane monooxygenase genes when you grow the organism on MTBE. But certainly the current evidence says that that enzyme is not responsible for MTBE oxidation, and nobody has ever been able to grow PM1 on propane despite efforts. And perhaps more currently with Pseudonocardia dioxonivorans, one of the model strains for 1,4-D degradation. We also see production of propane monooxygenase A, the expression of that gene during growth on 1,4-D. And we can also, there are reports of the upregulation of that gene when you take resting cells and expose them to propane. In this case, this organism, we know that 1,4-D is being biodegraded by tetrahydrofuran monooxygenase, which is what this gene is a component of. So, you know, we're left with this dilemma of neither of these organisms grow on propane. So what's propane monooxygenase, this PRMO, actually doing in these bugs? The last enzyme, and perhaps the one that we want to talk about for a little bit, is this last enzyme, perhaps the most recently discovered one, which is propane monooxygenase. I put it up as propane monooxygenase number 2. If you go back and think about how these enzymes are named, I'm not sure that calling it propane monooxygenase is necessarily the most helpful description. While it will oxidize propane, it's pretty clear that it will also oxidize quite a range of short chain alkanes. So as I indicate, for the purposes of clarity, we're calling it short chain alkane monooxygenase here with a distinctive name, SCAM. Historically, this was discovered back in the early 2000s, found in both Mycobacterium strain TY6 and that workhorse of multiple enzymes, Mycobacterium tubuense MBB4. Genome analyses have shown that it's actually all over the place. We find it in this Rhodococcus strain IFP 2016, which is an MTB degrader. Rhodococcus aethovorans BCP1, which is a chloroform degrader, amongst other things. We also find it in Envirogen or EMV421, which is one of the organisms originally isolated by Rob Stefan's group. What is the role, what is the significance of this enzyme? If you look at strain BCP1, we know that it grows on a large range of alkanes. When you look at a transcriptional study that was put out by in 2005, what's observed is that the hydroxylase component of this enzyme is consistently high for C2 to C5, C7 alkanes. And this organism also has a propane monooxygenase, one of these group 5 enzymes, which is only expressed at any significant level when the cells are grown on propane. So we end up with a situation where this organism has 2 different monooxygenases, but seems to express them at different times, depending on what substrate is being utilized. Interestingly, very recently, this enzyme found in Mycobacterium dioxaneotrophus, PHO6, has been shown to oxidize 1,4-D very effectively. So this gives you a sort of summary of the detection based on genome analyses of these multiple monooxygenases in propanotrophs. So here are the organisms here, the strains, this is the growth substrate range. And what you find is that SCAM, this group 6, is found in a wide range of these organisms. Propane monooxygenase is found in quite a few of them. And also, so is this particulate hydrocarbon monooxygenase, this copper-containing monooxygenase. So the $64,000 question, I guess that's not very expensive in these days, maybe the $64 million question, is what enzyme does what in these organisms? I think that's a very complicated question. This is very quickly go through another propane only. This is an unusual organism that was isolated, described a few years ago. It grows specifically on propane and then nothing else, and then C10 to C36 alkanes. It has got 3 alkane hydroxylases, 2 cytochrome P450s, a long chain alkane hydroxylase, and a propane monooxygenase. And if you look at this, these are the 3 alkane hydroxylases, these are the cytochrome P450s and the alkane hydroxylases. These are the substrates that the organism was grown on, and these are the transcript levels for the different enzymes. And what you see is that propane leads to the expression of significant expression of 2 alkane hydroxylases, a long chain alkane hydroxylase, and a propane monooxygenase. So the issue is what substrates are giving rise to propane oxidizing activity in these organisms, and why. And there's just sort of a quick summary of the soluble diion monooxygenases that we find in MBB4. This organism has got 4 soluble diion monooxygenases. 2 of them, this SMMO, soluble BMO, and SCAM, have the ability to oxidize alkanes. Another 2 have the ability to oxidize alkenes, and this kind of reflects the fact that it was originally isolated on an alkene rather than an alkane. So in the closing parts, let's just quickly go through the detection methodologies. I realize I'm running kind of long here, but I will get through them as quickly as I can. Stable isotope probing. As a technique that was originally established for hydrocarbon utilizing bacteria, principally methane oxidizing bacteria. So if you want to identify either the ability of native microorganisms to oxidize propane, or to potentially identify them by doing DNA SIP, this is a perfectly practical approach. C13 propane is readily available at pretty reasonable cost, but I would not suggest this is something that you could do out in the environment. The costs are prohibitive, so it's potentially useful for microcosm-based studies only. Compound-specific isotope analysis, CSIA. You can measure propane utilization in the field, so you don't really need to use this technique for looking at the gas utilization itself. It does have uses, for example, in understanding what happens in bulk transformations, perhaps it's been widely used in marine environments. But for our purposes, it's mainly used to look for evidence of biodegradation of contaminants, such as MTB and 1,4-D. qPCR is well established and very sensitive. The key question I think you've got to ask is, what is the relevance of the target gene to the process of interest? And I think this issue of having multiple monooxygenases in propane oxidizing bacteria brings this to the fore. The other thing that you need to recognize about qPCR is that it's not detecting activity, it is detecting the potential. It detects the gene, it does not necessarily tell you that that gene is being expressed. And the last technique that is sort of somewhat more nascent is something that we've been working on, is this activity-based protein profiling, which is essentially a proteomics approach that has the potential to detect, identify, and quantify active monooxygenases. I'm briefly going to go through some of these before I wrap it up. CSIA, a lot of marine studies have looked at using CSIA for looking at the oxidation of propane and butane. We haven't talked about the anaerobic oxidation of these gases, but they are readily, well, not readily, but they are well known to be biodegraded, often under sulfate reducing conditions. And so these are some of the studies that have looked at the fractionation for propane and butane under anaerobic conditions. And also under aerobic conditions, again, we're looking at marine environments primarily. So the study in 2007 looked at the enrichments carbon and hydrogen for methane through to butane oxidation by marine sediments. Also evidence from the Deepwater Horizon spill and that idea that propane may jumpstart hydrocarbon oxidation during that degradation process following the collapse of the well and the subsurface release of all those hydrocarbons. When you look at CSIA for MTB and 1,4-D, this is a sort of summary study from that Monica Rossell put together in 2012, looking at the carbon and hydrogen fractionation for MTBE by essentially pure cultures of microorganisms. And the point that she made was that, for example, GPO1, which expresses an alkane hydroxylase that can biodegrade, we can oxidize propane and can also degrade MTBE, gets lost in the signatures that are associated with acid hydrolysis, whereas some of the other alkane oxidizing bacteria like Mycobacterium vacui JOB5, they get lost in the signatures associated with anaerobic cultures. So certainly from the pure culture point of view, CSIA for MTBE is not particularly helpful. In the case of 1,4-D, we recently published a paper, Peter Bennett from Haley and Aldrich and others, looking at the carbon hydrogen fractionation of 1,4-D using the strain Rhodococcus ATC 21198. These represent the signatures that we see for the alkane grown Rhodococcus strain. This is the signature that we saw for Pseudonocardia tetrahydrofurans oxidans K1, which has got this tetrahydrofuran monooxygenase DMXO monooxygenase. So clearly we can distinguish between the alkane oxidizers and the tetrahydrofuran oxidizers in terms of the fractionation. There's a statistical significant difference between the enrichments that we saw for propane and isobutane grown microorganisms. This may represent the fact that this organism contains both SCAM and a propane monooxygenase. And I should also point out that this fractionation, these studies are done with very, very high 1,4-D concentrations, hundreds of ppm concentrations. And it may be that this signature is associated with the ability of one or both of these enzymes to oxidize 1,4-D, but it's unlikely in our experience that both of these enzymes would oxidize 1,4-D at much lower environmentally relevant concentrations. qPCR, there are primers available for a lot of these enzymes, SMMO, soluble BMMO, AMO, and PMMO, they're all available. Not necessarily sure that these are particularly relevant for understanding propane oxidizing bacteria in the environment, although they can obviously oxidize propane. P450s in general are very variable, so it's very difficult to come up with qPCR primers that would be particularly meaningful for understanding P450s as we currently understand them. And I would say the same is probably true for the ALK-B hydroxylases, alkane hydroxylases. When you look at propane, the hydrocarbon monooxygenase, the particulate hydrocarbon monooxygenase, I think there's clear evidence that both its role in propane oxidation and contaminant biodegradation. I think further studies is needed for this. It's only been detected in a relatively small number of organisms, and they're sufficiently different from PMMO and AMO that selected primers can be developed. There are, for the group 5 propane monooxygenase, primers already available. Again, I come back to this question about the significance in contaminant degradation. You know, a lot of the studies that, well, the few studies that have potentially answered the question about whether this enzyme oxidizes environmental contaminants like 1,4-D, trichloroethylene have been done for high concentrations of these substrates for prolonged periods. And other than NDMA, I'm not convinced. So part of the other problem is that we find this enzyme, genes encoding this enzyme in a lot of organisms with other propane oxidizing enzymes. Last of all, in the qPCR line, we have short chain alkane monooxygenase, this group 6 enzyme. There are primers available for them. They were recently published by Deng et al. For dioxonivorans. We have also been working with MI, and they have an assay, a qPCR assay based on this enzyme at kind of the beta testing level. I think Dora will tell you perhaps a little bit more about it. Just to give you an idea, we have analyzed some field samples from a propane biostimulation process. We saw very, very good overlap between the quantification from SCAM and propane monooxygenase, but always or pretty consistently saw that SCAM was, abundance was about 20% higher than propane monooxygenase. And I think that points to the possibility that a lot of organisms, propanotrophs, probably contain both SCAM and propane monooxygenase. And bearing in mind, we're not detecting activity here, we're just talking about gene abundance. If the abundance of organisms containing SCAM increases, you would expect the abundance of propane monooxygenase to also increase in lockstep, even though propane monooxygenase may not be catalyzing any really significant reaction in these organisms as far as contaminant degradation is concerned. The very last thing I talk about is this activity-based protein profiling technique. This works on the idea that we can supply probes to organisms that contain active monooxygenases. This is a symmetrical probe with an acetylenic functional group at both ends. The enzyme catalyzes the oxidation of this probe, leads to its inactivation, and we can then, now we've got a covalently attached probe to the enzyme, we can then react that probe with using click chemistry to either make the enzyme fluorescent, or we can use it to purify it by affinity purification, and then identify it through proteomics approaches. The key issue about this is, unlike qPCR, is the detection methodology requires catalytically active monooxygenases. If the enzyme isn't there or it isn't active, it will not be detected. Our current evidence says that we can detect virtually every soluble dione monooxygenase that we have looked at, AMO, PMO, and particulate hydrocarbon monooxygenase. And really the limit of detection is determined by the fluorescence intensity if we put a fluorescent tag onto the enzyme, and the amount of protein needed for an MS analysis. The fluorescence is not really an issue because these are intensely bright fluors that can be attached to them, and certainly as mass spectral analyses get better and better, the amount of protein that we need seems to go down and down. This gives you an idea of where we're going with it. So this gives you an idea of, this is an SDS page gel of a pure culture of an organism that's expressing a soluble dione monooxygenase grown on different alkanes. We see the same enzyme being expressed in all cases. We can look at whole cell fluorescence if we don't break the cells. We have worked on separating these cells by flow cytometry. I'm working on the ability to do it with FACS. And that fluorescence then can be correlated with an activity. And so in this case, we're looking at the activity of these alkane-grown cells versus propylene oxidizing activity. So this is something that we think holds a lot of promise for detecting these type of enzymes in the future. So my final thoughts, I know I probably kept you here way too long, apologize for that. So what are they? The constellation of propane oxidizing enzymes, it's expanding. It's just like the universe. There are certainly over the last few years, this has certainly been powered by genome-based analyses that more and more enzymes are being recognized with the ability to grow, to oxidize propane. And there may well be more enzymes on the horizon. I think the neglected area is the cytochrome P450s, which we know can oxidize propane, but really have not been studied in any great detail. Certainly, I think from a practical point of view, the term propane monooxygenase is ambiguous, and it gets bandied around a lot. And as we know more about these organisms, these propanotrophic organisms, I think it is a term that should probably be restricted to the group 5 propane monooxygenases, which have this PRMA ABCD configuration. Having several enzymes called the same thing that do substantially different things and have different substrate ranges, I think is probably not helping much. Certainly automated genome annotations are not really helping clarify the situation about what enzymes are present. Finding enzymes that we would describe as SCAM, but are actually more conventional group 5 propane monooxygenases is something that the automated annotations haven't sorted out. And the same is true for the particulate hydrocarbon oxidizing monooxygenases, which look very like AMO and PMO. So you have to delve in kind of carefully into the genomes or metagenome type analysis to make sure you're getting the right enzyme. I think one of the biggest research challenges out there at the moment is to understand what the roles of these multiple propane oxidizing enzymes are when they occur in a single microorganism. And obviously that has impacts on understanding, for example, the signatures you get in CSIA analyses with pure cultures. It obviously has an impact on what enzymes or genes you want to detect in the environment. You may detect one enzyme thinking that it's representative of propane oxidizing activity that you think, but it's actually not. The enzyme doesn't do what you think it's doing. And lastly, I think the analytics are going to start to emphasize proteomes over genomes because, again, of this issue about having multiple enzymes, being able to identify which ones are actually being expressed as opposed to simply being present and whose numbers are increasing is going to give us more and more resolution in the environmental field. And I put in this last plug here, if you want to have a, with a no promises attached sample that we're interested in looking for these APB analyses, please feel free to contact me because we are very interested in expanding the use of this into environmental field. Lastly, thank you very much for joining today, and I'm sorry I've run a little bit long, and I'd be happy to answer any questions.
All right. Thank you very much, Mike. This was a really interesting and informative presentation. And just as an aside, we have uploaded a one-page handout that you should see on your GoToWebinar control panel that talks a little bit about the different assays that are available that Mike discussed during this webinar today, so just as a helpful aside there. And then if you're interested in having a copy, a PDF of the slides, Mike is going to put them together and add a bunch of references that may be useful to you. It'll be about a week before we have those available, so if you can be patient with us on that, send us an email to info@microbe.com requesting that you'd like a copy, and we will send that out to everyone on the list once those are available. Okay, Mike, we got a couple questions here that look good. Can you talk a little bit more in depth about distinguishing the difference between the soluble and the particulate on the monooxygenase genes? What does that mean in particular? And does particulate enzymes meant to imply it's attached to the cell?
Oh, okay. So particulate, let's ask the last part first. So particulate means that the enzyme is membrane bound. So in other words, if you go through the cell from the outside first, you will deal with the cell wall, whether that's a gram-positive or a gram-negative cell, it doesn't really matter. The next membrane that you will come to is the cytoplasmic membrane, which really represents the delineation between the inside of the cell, where all the soluble enzymes are, for example, where all the soluble diion monooxygenases are, and the outside world. So particulate enzymes are membrane bound, and they are present in the cytoplasmic membrane. So they are, location-wise, they're very, very different than the SDIMOs. Structurally, they're very, very different. They tend to have a lot of transmembrane helices. So the protein is deeply embedded in that membrane. So I think that qualifies or hopefully explains the location issue. What was the first part of the question?
The first part of the question was just different. I'm sorry. The first part was just how do you differentiate between soluble and particulate? What's the main difference between the two?
Well, activity-wise, it turns out that perhaps it's less clear than you might think, because as I've probably illustrated it, overindulged in explaining that the substrate range of these enzymes can overlap quite significantly. There's the cellular location. Certainly structurally, they are very, very different. So all of the particulate enzymes have this, the major ones that we talked about at least, AMO, PMMO, and PHMO. They all have this CAB structure, operon structure. So that's very, very distinctive. The molecular weights of the subunits are very different. The types of subunits that you have are very, very different than the soluble enzymes. You know, they really are, should not really be put in the same category as the soluble diion monooxygenases, except that they catalyze some very, very similar reactions.
Okay, that's really helpful. The other question that we have, Mike, is about 1,4-dioxane, and I think maybe I know what he's referring to here, but the question says, have these methods been able to get down to the New Jersey groundwater standards to 0.4 ppb? So I think maybe what he's wondering is, you know, about degradation of these compounds in very low concentration. Okay. Are we able to, under monitored natural attenuation, be able to do that?
I think that's a very, very important question, and it's something that I think I hopefully alluded to. There was a slide there, I think, where I put up, you know, certain bugs could degrade TCE, certain could do MTBE, certain ones could do 1,4-D, etc. What you have to bear in mind is that in a lot of cases, you can force these enzymes to exhibit activity for these contaminants if you just conduct pure culture studies and you load up the concentration of substrate like 1,4-D. And a good example of that would be, for example, there was an early report that methane oxidizing bacteria would oxidize 1,4-D. If you look at the original description of this, these analyses were done with concentrations of 1,4-D at about 500 ppm. That's a far cry from the 0.4, the 0.5 ppb that that you're talking about for this New Jersey standard. And it turns out if you start looking at methane monooxygenase and examining much lower, more environmentally relevant concentrations of 1,4-D, it's very difficult to find that methane monooxygenase, SMMO, will oxidize 1,4-D. We've had the same experience with MTBE. If you really load up the concentration of MTBE, you can get soluble methane monooxygenase to degrade it, and you can determine that by the detection of tertiary butyl alcohol as a product. But this is at concentrations of thousands of parts per million of MTBE. So that's why I put the caveat on that slide that, you know, the concentration on which these assays were done and the corresponding ticks that you put on that chart is very dependent on both the concentration of MTBE or contaminant that you're interested in and also the length of time of exposure. You know, I think a reasonable assay for looking at spontaneous activity for an enzyme might be if you grow the cells up and ask what does it do over a matter of 2 or 3 hours, but doing it for 28, 48 plus hours, you really have very little understanding or control over what the cell is actually doing. At the other end of the spectrum, I think you're going to find that there is the same would apply for the evidence for propane monooxygenase, PRMO, oxidizing 1,4-D. I think that certainly our experience is that enzyme organisms that express SCAM can oxidize 1,4-D at extremely low concentrations and very likely to the concentrations that your questioner was asking about.
That's very helpful, Mike, and we really appreciate you taking the time and doing this great presentation for us today. It's a really good overview, and I think the slides are going to be really helpful to everyone, so we appreciate your time so much on this. I think that's all the questions that we have for today. If you have additional questions, you can email Mike, as we mentioned earlier. You can also email us here at Microbial Insights, and we'll get with him and get the answer for you. We appreciate you all taking the time out of your busy schedule, and we appreciate you, Mike, for taking the time to educate us on this really important topic. I hope you all have a wonderful day and have a great rest of the week. Thank you so much.
Yep. Thanks.
Bye-bye.
Appreciate it.
Thanks, Mike. Bye-bye.